many of us. And it's so wonderful, the hospitality and the science. Don't even talk about the food and a lot of alcohol after the conferences. So basically, I would say, it's a spirit of work hard, play hard. So we got to really discuss the great science and also the fun friendships. I think this is basically, it's the go-to meeting every couple of years. So we're looking forward to it. So unfortunately this year, due to the Mr. or Mrs. COVID visits to globally, so we cannot do this in person. But thanks to Dr. Ang's persist, so we still doing this virtual, although not as ideal as in person, but this also give us the opportunity to get to interact and talk to each other and share science. And we just realized, and that also has a CBI logo on the speaker behind. Sean, where's yours? And so it's really nice, and hopefully, and then I also just realized James actually working together with my instructor Shima. So we were on the email in the past. I did not even recognize that. If not because of this meeting, I will never know and remember. Now I will remember. So this is just a wonderful opportunity, for everybody to be gathered, talk about science, talk about career, talk about families and everything. Even earlier, Sean, you did not heard that they are asking about how's Joy. I think Joy is my neighbor. I can actually knock on the door, bring her here. So it's really wonderful. So, and I think with this, so it's by recording. So Dr. An, it's currently a professor emeritus in the Seoul National University. And his research field is it's cutting edge semiconductor at the beginning of the semiconductor, and including the thin film transistors and for various applications. So he actually, started like many of the panels here, speaker here is they start his professional career at actually UT, the US, and then is a system professor in electrical computer, engineer at the US and in New York actually. And then, in between, he also returned to SWORD and to establish, a different things and being a dean of College of Engineers. And then, he really has a lot of inventions. And I think that maybe also build his careers and allow him to really contribute to, many of those technology fields. So he really is one of the amazing people, and it really exemplifies what a successful, physician scientist and scientist should be. And so with this, I think we have an honor to really got his vision points to us via video recording.
Good afternoon to the distinguished speakers and participants joining from Stanford University, the United States. And good morning to the distinguished speakers and participants from Jeonnam National University, Korea. The Lunar New Year is approaching, and I would like to wish you happy and prosperous New Year. Today, I'm very much pleased to welcome you to the 44th CAST International Symposium entitled Advanced Therapy in Cardiovascular Regeneration, Korea-Stanford Joint Symposium. Today's symposium is part of a series of academic meetings held since 1999 by the Korean Academy of Science and Technologies. The CAST International Symposium aims to provide a good opportunity to exchange valuable scientific achievement among renowned scientists from all over the world. It is unfortunate that we can afford to meet you in person due to the COVID-19. Of course, we may miss a glass of wine after face-to-face symposium, but I'm still thankful that we can continue our academic journey online. We also don't have to waste so much time and money at the airport and on airplane as well. Thanks God. Today, my special gratitude goes to Professor Young An for his superb efforts in organizing this symposium. Thanks to Professor An, we are honored with presence of cardiovascular science experts from Korea and the United States. I hope today's symposium not only provide the exchange of research findings, but also strengthen the network among the participants for future collaboration. Let me now close by wishing you a delightful and fruitful symposium. Thank you.
But I think it's he has a message for the warmest welcome to everybody to join in the Stanford and Korea some joint cardiology meeting. So I am Young-Nan from the Jeonnam National University Hospital, and I'd like to start in a session first. We have 3 speakers. So after presentation, we'll have a discussion with the panelists and also from the audience. So let me introduce in the first speaker, Longlin Liao from the Stanford University, United States. I think it's I don't need to some general some description of the Longlin because she is world famous. The reading the researcher about in the especially some the elucidation of the mechanism, key mechanism of from the normal heart to heart failure, and then how can some the reverse this from the heart failure to the normal heart. So on today, she will talk about the impact of tobacco smoke and the stress in cardiovascular health. Longlin, please.
Thank you, Dr. An, for kind introductions, and it has been the honor to get to know you, and we become a great friends. And thank you for always think of me and giving many opportunity to invite us to visit Korea. I just mentioned that was really wonderful experience. So today I want to share with you a couple of projects that we actually started not too long ago at Stanford. Although, some of those projects may be, still kind of a preliminary. So, but I thought it's really very fitting for this current situation of global COVID pandemic. So I'm certain that all of you all agree this COVID pandemic really impact internationally, and it's a rather stressful condition as we're just talking about the stress. And we have to change in this conference to a virtual conferences. So as I mentioned earlier, visiting Korea to attend these conferences and seeing our Korean colleagues and, and then the friends there for a long time in person, I think this is one of the most exciting events. I mean, I certainly enjoy it. I'm sure I can speak for everyone. We all anticipate it. But I'm sure I'm not the only one feeling disappointed, as this COVID pandemic is really, add much stress in our daily life. And to make it worse, I believe many people actually deal with the stress by consuming excess of alcoholic drink. Okay, so when we go to Korea with a dinner, excess alcoholic drink, that's a different content. But normal content with a stress, you have an alcoholic drink or a cigarette smoke. And ideally, it's people with intention to relieve stress. But however, drinking or smoking, it's not only could not reduce the stress, it actually could worsen our health and even adds on the stress. So, according, you see the statistic here, according to WHO, more than half of the Koreans are smoker. And recent reported the data also show that smoking worsened the COVID outcomes. And so today I'm going to focus on our unpublished data on the impact of tobacco smoke on the cardiovascular health. So impact of tobacco smoking on the cardiovascular health, like I was saying, it's a new topic and a project to us. But I believe this research topic is especially relevant to the current COVID pandemic. So that's why I would like to share with you some of the initial results from two new and ongoing studies. So the first topic is the research led by Dr. Chen-Yu Tsai, a brilliant postdoctoral fellow joined our lab a couple years ago, and he actually just returned to Taiwan to start his own independent research. So Chen-Yu is interested to investigate how could psychological stress in combination with smoking impact the heart health, so cardiac health. And we often hear people or ourselves saying, I'm really stressed. In fact, stress is not just an emotion. Stress could actually actively affect our health. And as you know, many people deal with the stress by consuming alcoholic drink or smoking. But unfortunately, smoking or alcoholic drinking actually serve to increase stress level further rather than release stress. So shown here are a few publications showing that stress could indeed lead to the increased tendency of cigarette smoking. So unfortunately, all the tobacco products contain nicotine, which is a highly addictive compound. That's why smokers, sometimes they feel stress relief, and during the time they smoke. But they did not realize that smoking actually triggered the entry of a vicious cycle that leads to the development of a nicotine addiction. And so that the smokers, while they understand smoking may not be good for them, but they still rely on increasing smoking to deal with their stress. And then, of course, that comes with the negative impact of the cigarettes on their health. So unfortunately, if we search the literature, it's very limited study focused on the impact of stress on our health, especially the cardiovascular health. So when we are under stress, our body actually produces cortisol, which is the primary stress hormone. Therefore, what the smokers experience is not just the nicotine, but also the elevated level of a circulating stress hormone. Although it is well known that nicotine can induce addictions and cause cardiovascular disease, however, the combined effects between nicotine and stress hormone are poorly understood. So using the animal model, Chen-Yu asked, if the stress hormone level serve us to alter the nicotine consumptions. So to answer this question, Chen-Yu delivered corticosterone to mice, and that's actually the stress hormone in rodents, by daily injection for 2 weeks, and then house them individually with 2 bottles of the water in the cage. And 1 bottle contains the regular drinking water, and the other contains the water contain nicotines. And by measuring the amount of water consumed from each bottle, we can then calculate the nicotine preference level. So remarkably, our results show that the mice injected with stress hormone actually has much higher nicotine consumption of the water content nicotines. And so increase the nicotine preference compared to the mice inject with saline alone. So this result really demonstrates a strong relationship between the stress hormones and the nicotine consumptions. To further investigate if the nicotine consumption and stress hormone, either alone or in combination, affect the cardiac functions, we separate them in the several groups, as shown here, the nicotine alone group, stress hormone alone, and also the in combinations, and of course the saline control. And then we do this in a 6 weeks duration. As you can see here, cardiac functions and then structural remodeling is determined using the non-invasive ultrasound image, as everybody has been using it. It's very nice and common measurements for the functions. And we decide to using the strength analysis because this is highly sensitive and then can detect the signal of early dysfunction. So our results showing that only the mice treated with both nicotine and stress hormone show decreased the cardiac function after just 1 month exposure to nicotine. And in addition, our results also show that the cardiac functions alteration is accompanied with the increasing wall thickness. So it's not just a function, also has a structural remodeling. And, and also the increasing of the transcription factor of MFC3 and GATA4, which are the transcription factor known to play an important role in the pathological hypertrophy. So the addition resulted from the cigarette smoking may explain why the smoker continue to smoke, albeit they most likely aware of the profound negative impact of smoking on their bodies. And, on the men, even worse than the females, and showing here, they led to our first question. Is there any direct relationship between the stress and the nicotine consumptions? So this study is still ongoing. So it is important to point out that smoked animal for 6 weeks is only equivalent to human smoking for about 4 years. But however, based on the statistic, the average smoker is about 20 years smoking history. So therefore, to truly modeling the condition in humans, we are actually cooking some animal right now and then to make them smoke longer durations in order to really determine the true impact of smoking in the cardiovascular health. So the second story is conducted by another very talented postdoc, Katerina Sumot. Katerina is interested on the impact of cigarette smoking during pregnancy on the regenerative capacity of the offspring. So this including it into it is because this section we're supposed talking about regeneration. So here you go, some regenerative data here. So it is well accepted that adult rodent hearts, much like human, there's a very limited regeneration capacity following myocardial infarction. And the heart muscle will die and replaced with scars. And, as we are well known, shown here in the figure. So on the right-hand side panel here, it's showing in contrast to the adult hearts. Neonatal has much robust regenerative capacity as shown here. They can fully recover from their cardiac injury from the myocardial infarction if the injury is posed much early on in life. So this is something, I don't need to continue to talk about this because everybody knows. So what is shocking to us is that 1 in 14 women still continue smoking during pregnancy. And so Katerina is interested to figure out if smoking during pregnancy impact cardiac regeneration of offspring. And so Katerina subject the pregnant mice, the mother, to a cigarette smoking using the smoking machines and during entire pregnancies, and using this commercially available smoke machine is pretty cool. so we can smoke them. And then strikingly, we find out 7 days after the induction of myocardial infarction, the hearts of a neonatal mice born from the mother exposed to the cigarette smoking during the pregnancy has very limited capacity to recover. So those pups undergo similar deleterious structure and functional remodeling, much like adult hearts. So this data is really suggests that neonatal cardiac repair is lost, while that non-smoke litter mats still maintain their regeneration capacity. So this really call out the importance of the deleterious effect of smoke to the neonatal health. So I think, this, it's really summarized. It's important to really send out the message to the smoking mother. So our data suggests that, this is really very a significant negative impact to the, the regeneration capacity. So we believe that it is important we really advocate this message to the fields. So with this, I wish to thank the various sources of our funding agency for our research, and as well as our remarkable research team for the generated data and doing all the hard work. They are a group of very talented fellows and a young investigator and future superstars. And talking about future superstars, we are currently looking for motivated postdoc or visiting scholar to join our research team. If you are interested, please email me. And last but not least, taking care of our hearts and build a smoke-free society and free of cardiovascular disease. And so again, thank you very much for giving me this opportunity to share with you our results. Thank you.
Thank you, Rong-Mei. So I think we will have a discussion at the end of the session. Can I join with the postdoc fellowship with your lab next year?
You will be a professorship, not a fellowship.
Yeah. So that's
I need to stop sharing, right?
Let me introduce in the next speaker, Shanu. Also, he is also my best friend. And then actually, I also don't need to introduce in his whole story, but his world reading famous in the researchers, especially in developmental and the stem cell biology of the cardiovascular system, and apply these findings to repair and regenerate the diseased heart. So today, he will talk about the leveraging mechanisms of cardiomyocyte proliferation for regenerative applications. Shanu, please. Sean, so please, okay.
Yeah, okay, so can everybody hear? I hope so. Yeah, I want to echo what Rong-Li just said and want to really thank Professor An for the invitation to speak at this meeting, and also thanks Professor Young-Soo Kim for organizing all the speakers. And it's always such a wonderful time interacting and meeting with everyone in Guangzhou when it was in person. And I completely agree, Rong-Li, that there's just so much more friendship interactions during the time in those, Guangzhou meetings that I really miss this, and hopefully we can return to doing those meetings again, when this COVID visit eventually goes away, and hopefully very soon. And so what I'm going to do is to talk about the work that we have been doing recently in understanding some of the cardiomyocyte regulation that it affects proliferation. So I'll go ahead and do the full screen share. So I'll start from here, which I think everyone is very familiar with, and that is what happens when you have a myocardial infarction from the usual coronary artery closure. And what you could see histologically is that a normal heart has nice pink healthy tissue, and a diseased heart has a significant scar regions, and these scars are largely irreversible. And then so the fact that this particular region is no longer able to provide the contracting function leads to the subsequent remodeling and eventually the failure of the ventricle. And over the years, I put this slide here to sort of give a little bit of perspective from the therapy side of what people have really tried to work on for this issue, ischemic heart disease. We know that we started with a lot of drug treatment, things like beta blocker, ACE inhibitor, which has now become the mainstay therapy for heart failure. That's usually for people who are at least still able to compensate to provide normal amount of flow to the rest of the body. But in people who have extreme heart failure where the heart no longer support the perfusion, cardiac transplant is the extreme end of the therapy spectrum. But we also know that the number of hearts available is limited, that not everyone who needs to have a new heart is able to get one. And then so over the years, there's been a big effort to try to fill this large gap in therapy between these two spectrum. And then so some of the things that people have been looking at, things like biologic therapy with either peptides or antibodies, have been looked at as a therapeutic agent. Those are continually being explored. But what has become more successful is the mechanical therapy. So the treatment of using things like the ventricular assist devices have become probably the major mode that people are receiving the heart support. And so these therapy, which requires a lot of cost and a lot of invasive procedures, have come with lots of potential consequent side effects, such as thrombosis and infection, that have still remained to be quite challenging. So because of that, we continue to look for new ways to treat heart failure due to MI. People are looked at using gene delivery, such as with the AAV9 for correcting heart function. Those are still being looked at. Cell therapy has been a real topic of interest over the decades, and many people have tried a variety of different cells as a way to do therapy. And then some combination between genes and cells are also now being considered, given especially with the way of editing cells in the genome. And then so all of these things right now are in the exploratory phase. And then so the focus that I'm going to talk about is cells as therapy. And then so this slide describe a little bit busy, but it sort of covers the various different ideas of what kind of therapies there might be for repairing a damaged heart. So the top box in pink shows what might be some of the cells in the heart itself, endogenous cells, and what kind of methods to stimulate these cells might be able to provide a way of expansion and eventually the therapeutic aim of these different cell types. The endogenous cell population has been a little bit challenging because the potential for them to become cardiac cells is also a little bit less than the cells that you otherwise may be able to derive from other sources. And then so the exogenous, so the extrinsic to the body sources, such as cells from the bone marrows, or cells derived from pluripotent stem cells, or even isolating a cardiac stem cell from another person. These therapeutic strategies have also been looked at over the last 10, 15 years. And again, I think a lot of what people have found is that we were able to induce some beneficial effects, and possibly through a paracrine growth factor related mechanism, but in general, that still remains to be seen how much of an effect we can achieve and whether we can enhance some of that to get better response. There are, on the experimental side, certainly approaches like directly programming the cells, like taking a fibroblast that's either within the heart or isolated outside in culture and introduce different transcription factor or lineage programming factor and try to directly convert cells either into a cardiomyocyte or into progenitor cells. So all of these strategies are actively looked at by many different investigators. And the part that I'll focus on for this talk is the work related to using a pluripotent stem cell source, such as embryonic stem cell or induced pluripotent stem cell. I think a lot of the reason why the embryonic and pluripotent stem cell has become of interest is because you're able to generate quite a large number of cardiac myocytes that are able to beat readily in the dish. So that gives people a lot of enthusiasm that we have a replacement source to be able to provide this lost cardiomyocyte. But there are also some, issues when it comes to introducing these cells for therapy. And then I list some of the challenges that people have been working on over the years. So the first issue I think everyone knows is there's always going to be possibility of toxicity or risk. So the safety issue is extremely important. Is it possible that we can transplant and put the cells into the heart that doesn't involve introducing tumors, either from the cells that we looked at? Is it possible to have it in the heart during injection without excess injury or bleeding during the introduction? So these are all some of the issues that people have spent a lot of time working on. And then so one of the things that say that this issue of safety is able to be overcome, there's also the efficacy part, and the efficacy both involves engraftment, how much of the cells that gets put in is actually able to stay. Of the cells that do get to stay, how much can they actually expand? Because most of the cardiomyocytes are terminally differentiated, so they don't really expand very much. So is there a possibility of expanding them afterwards? Because otherwise you have to start from the cells that's the most primitive in order to get to the biggest number. And that may be quite challenging to identify the cells that's appropriate. And then the cells, once you introduce, is going to have to mature, further differentiate. And will they be able to really further differentiate and mature into the cell of interest? And then once they become something that is a true cardiomyocyte that's mature, they also then have to integrate and connect with the existing cardiomyocyte so they can all function together. And there's been a lot of issues with the cardiomyocyte that are injected, but they're not necessarily integrated and causing arrhythmia type of issue, which I'll comment on in a second. And then the proper enhancement of function after it does get introduced is yet another issue. And I think we're not even quite at the point of figuring out what kind of issue with the rejection and ultimately the cost for these therapies. So again, a long road to go, but I think because we see very limited other options at the moment, these are still going to be research topics that will be actively investigated by many people. So I want to just comment on a couple papers that I think has really driven the thinking in the field. So Chuck Murray's group has done a lot of injection into the non-human primates. And in this study, they used human ES-cell-derived cardiac myocyte to inject into non-human primate. And you see that these yellow greenish area represents a human ES-cell-derived cardiomyocyte graft are probably the largest graft that anyone has really seen to date. And these are, so to give some potential promise that this may actually be a strategy that works, but yet, one of the issues, and they, along with the former postdoc, Yuji Shiba, who after he went back to Japan, used iPS cells to show very similar engraftment, is that in both of these models, there was significant issue with toxicity. So the animals all developed ventricular tachyarrhythmia, even while they were being treated prophylactically with amiodarone. So it suggests that there's a lot of issues with the cells and their phenotype and potentially their ability to truly integrate that's causing the arrhythmic problem. And then so this really does require a lot of study repeatedly with lots of different conditions in animals, but because the studies were done injecting on the order of 1 billion cells, to get to that number of cells, really takes like an entire village, according to Chuck Murray. And this is something that's very difficult for the people who have worked with iPSL, is to how to get a lot of cell number. So we've been looking at using the pluripotent stem cell, particularly iPS cells, as a source of treatment. But as I just mentioned, because the work is very labor and time intensive and costly, and every time when you do these differentiation, there is batch-to-batch variation, there's been a lot of interest in trying to figure out how do you get even more cardiomyocyte every time when you do this differentiation. And that's sort of where we started the work, is trying to figure out how do you scale up and expand. And if you can get the cardiomyocyte to scale and expand, there are many different uses, such as disease models using cardiomyocyte from iPS, or screening drugs for therapy, and assaying for toxicity issues, and potentially even things like clinical trials in the dish in humans. So, we believe that this is an important, area to address in figuring out how to expand cardiomyocytes. So I wanted to just sort of divert a little bit of the attention, because the rest of the talk I'm going to mention relates to the signal pathway, which is extremely well known and involving many different processes. So that's the Wnt signals. I think many people knows that the Wnt signal involve various components, such as the Frizzle and the LRP receptor, who are the upstream of a downstream complex called the axon destruction complex, involving different players such as glycogen synthase kinase, GSK3, and beta-catenin. And when the Wnt ligand engages on the Frizzle receptor and the LRP, what it does is it pulls up this axon destruction complex and frees the beta-catenin from degradation by the proteasome ubiquitin process, so that the free beta-catenin is able to bind to the cellular target at the TCF loci and induces the Wnt target gene. And more easily, instead of using the Wnt ligand, many people now directly inhibits the GSK3 in order to get the effect similar to Wnt signal. And that involves using a compound called CHEER 99021. And I'll refer to that as just CHEER itself. And so early on, what we have found, so this is a paper from Jan Bukema, who became a postdoc in my lab, but while he was doing his PhD training at Mass General Hospital, where I met him at the beginning, he was looking at a way to modulate Wnt signal. And what he found in this paper is that if you reduce the function of Wnt by the loss of function Wnt model, you reduce the myocardial thickness and the cell number. So we know that the Wnt signal is required or necessary for cell proliferation. But the other thing that was also interesting was that if you increase the Wnt signal, there does not appear to be a great enhancement in the myocardial thickness. So suggesting that while Wnt is necessary, Wnt alone does not drive the proliferation. And so that's sort of an important concept that I'll focus on in a minute. When other investigators then tried to look at the Wnt shear signal in proliferating cardiomyocyte in vitro, say using pluripotent stem cell, they show that there is, again, that increase of a stimulated proliferation treating with shear, but in none of the cases do you get an extremely large number of cardiomyocytes. So the number of cardiomyocytes may be increased by 30, 40%, but not necessarily dramatic. So not exactly the amount that you really need to have if you want to get a lot of cardiomyocyte. And then so what we wonder was that if you have cells that are growing and in the dish, as they continue to grow, they eventually will become confluent. And when the cells are confluent, there's a very well-known described phenomenon called contact inhibition, such that the cells in contact reduces their ability to proliferate. And then so what our hypothesis was that forming of these cell contact would inhibit the iPS cardiomyocyte to proliferate, despite when they're in the presence of Wnt signal activation. And then so with that idea, then we went through and studied this process of the Wnt activity and cell contact as a potential synergistic activity that it may be regulated to proliferative gain in cardiomyocytes. So in this paper, which was published a few months ago in Cell Stem Cell, and from the work of Jan Bukema, who was a postdoc in the lab, who's now back in Netherlands as a cardiologist, almost finished training, and also So Lee, who's the current postdoc and senior postdoc, who is also on the job market. So for any of you in Korea who's looking for new faculty, please definitely reach out, because I know Soa is going to be very enthusiastic about talking to you for any potential opportunity for faculty position. So what the two of them have looked at is whether or not in the presence of this GSK3 beta inhibition, so Wnt stimulation, and regulating the contact activity between cells, you can get a massively expanded cardiomyocyte population. And so what Soa had done is what she took the iPS cardiomyocytes that you normally get, differentiate into the different days until 12, and then test either by not passaging at all or passaging, but still maintaining very dense population. So maintaining cell contact versus sparsely passaging, so keeping them very loose and not in contact. And what she saw was that there's a significant, and this is the log units, just to remind you, of the gain in proliferation cell number when you sparsely culture, rather than either not passaging or passage, but still keeping the cell density enough that cell contacts are present. So this really tells us there's a very significant inhibitory activity when the cardiomyocytes are touching. And this immunostaining confirms the increase in Ki67 level in the cells that are sparsely passaged. And so with the continuous culturing and expansion in the presence of Wnt signal and serially passaging to keep the density low, we were then able to get multiple rounds. So you can see one small dish of cardiomyocyte can give you tons of cardiomyocytes in the dish in the end. So this has become very easy to do in the lab to get, on the orders of hundreds, million to billion cardiomyocyte on the regular basis. And one interesting advantage we also saw is that when you treat with this cheer and passage, you actually maintain, if not slightly enhance, the purity of the cardiomyocyte, as opposed to if you do the same thing without the Wnt stimulation, eventually the non-cardiomyocyte takes over and the fraction of cardiomyocyte actually drops significantly. So that I think was a nice surprise that we had not necessarily expected. But we also showed that this treatment effect increases the folds in the orders of 200 to 300 and is able to do it in multiple different lines. So these are 4 different lines in the Cardiovascular Institute that we are able to gain the proliferative gain. So, quite a robust and reproducible and consistent effect. So one of the question we had is the YAP signal, which is well known to proliferate cardiomyocyte, is involved. And we saw that even though what Jim Martin has shown that these the YAP signal, when you regulate the pathway by knocking out the salvo inhibitor, is able to get a huge cardiomyocyte expansion in the neonatal and adult hearts, we were not actually able to see an increase due to YAP in our model. So here, by inhibiting the YAP signal with verteprofen, we're actually getting we're inhibiting YAP, but instead of getting no proliferation, like one might expect when you inhibit YAP, we're actually got an increase in proliferation. And what we noticed was that the proliferation was accompanied by 2 features. One is that the cells are increasing the spindle-like shape with protrusions, and then the other is that the sarcomeres are completely disorganized. And the sarcomere disorganization part was quite interesting because we then looked at the data that we generated with single-cell RNA-seq to find out that if you sparsely culture the cells, the genes that are regulating sarcomere organization and myofibril assembly decreases. So the genes like TCAP and leomodin-2 reduces when the cells are sparse compared with dense. And this was consistent with a very recent study from Didier Sagnier's group on using zebrafish model, that inducing sarcomere disassembly and increasing these spindle myocyte protrusion during zebrafish regeneration is able to promote the proliferation. And then so in their zebrafish model, they show this protrusion extension by reducing sarcomere assembly as a way to increase proliferative gain. And we also saw the same thing with the Wnt model in iPS cardiomyocyte, that when you treat with a Wnt stimulator or GSK inhibitor cheer, you get this very disruptive sarcomere with increasing nuclei for the dividing cell. But fortunately, this process can be reversible. So if you treat it with cheer and then hit it with the C59, which is inhibitor of Wnt, they can actually reorganize the sarcomeres to make them align. So the reversibility is actually very nice. And then so one final slide that I want to present is that one of the question, if the cells are disorganized when they proliferate, will they be able to function if you stop the proliferation? And then, so in this experiment, we generated engineered tissue where we first expanded the cardiomyocyte to day 42, but then we let them further mature for another 25 more days in the engineered tissue format. And in this format, we saw that the sarcomere was able to realign and organize over time, and that when we measured the non-expanded to the expanded cardiomyocyte, we get pretty similar, if not even increase, in the fourth generation in the engineered tissue. So we're glad that these cells actually are just as functional as the ones that we normally produce without the expansion. So in summary, I mentioned to you that in the adult heart, which rarely regenerates, has very limited capacity for repair after injury, but that we, by looking at the Wnt and beta-catenin signaling and combining that with the reduced cell contact, is able to induce a massive expansion of the iPS cardiomyocytes. And we show that the cell-cell contact inhibition of this is not due to a YAP-mediated signaling event, but it does involve the promoting of the sarcomere disassembly as the mechanism for increased myocyte division. And when we do the single-cell RNA-seq study, we confirm the reduction of these sarcomere assembly gene when we disrupt that cell-cell contact. And then finally, we show that when you then engineer heart tissue from these iPS cells after they've been expanded, they continue to be able to contract with similar force as the unexpanded cells. So, you know, the functional activity of these cells are retained after expansion. So with that, I want to thank the folks in my lab. So I highlight So Elise, as I mentioned, she's intended to find a faculty position after completing her training with us, and also Jan Boekema, who's in the Netherlands, finishing his cardiac studies. And we have lots of collaborators from different places within Stanford, as well as outside in various institutes, who have really helped us with these studies, and also the funding sources that supported various different research work in our research lab. But last not least, I want to thank everyone for your attention, and would be happy to take any questions.
Okay, thank you. So we'll have a discussion at the end of the session, and then I think it's really wonderful your some research product. So congratulations on your recent, it's a very tremendous some results. So I will introduce in the third speaker, Hwang Hyun. Actually, he is my colleague in my university. Actually, I just briefly introduced in the him. He graduated Jeonnam University Medical School in 2007 in Korea. And then right now he is the associate professor in Jeonnam National University Medical School in the Department of Pharmacology. Right now he is the visiting assistant professor, University of Pennsylvania, the Perelman School of Medicine. So today he will talk about the PSME4 degradate YAP1 of mesenchymal stem cells in the nucleus to induce cardiac commitment. Dr. Please.
Thanks, Professor Han, for your kind introduction. I'm very happy to see you again in this COVID area. Yeah, COVID also affect my lifestyle, especially to my hair. Okay, I'd like to talk about the collaboration project with Professor Han's lab. The title is, here. Even in South Korea, the cardiac disease is a leading cause of death. It is second highest cause of death. When you took a look in 10 years history, the people from ischemic heart disease only increased just at 6%. But the heart failure, the almost dramatically increased, almost doubled. Because the physician's skill and the post-event treatment strategies well established, mortality of ischemic heart disease was not so much increased. In spite of patients' effort, about 30 to 40% of ischemic heart disease patients experience heart failure transition. Recently, the total number of deaths from heart failure in Korea is much more than ischemic heart disease. And why ischemic heart disease results in heart failure? The ischemic heart disease, in other terms, acute myocardial infarction, was developed by acute loss of coronary circulation. Many of the cases are generated by blood clots, which are made from in situ or the proximal lesion. This impaction of blood clot blocks the blood circulation in the distal myocardium. Yeah, because cardiac blood circulation lacks peripheral supply, distal part will die. Our body cannot replace the myocardium. This finally results in global remodeling shown here. So many scientists have tried to replace the myocardium to a fresh, live myocardium in parallel of minimize of myocardial death. Among various trials, the stem cell-based regeneration project is still attractive and promising. So the tons of studies try to prove the effectiveness of stem cell therapy. Still, we don't have clear evidence how delivered stem cell improve cardiac function. As a possible mechanism, stem cell itself barely differentiate into the cardiac cell. Stem cell produce many beneficial cytokine and minimize myocardial death. This cartoon shows the long history of a stem cell therapy strategy. At first, various sources of stem cell is directly delivered to the infarct area. Somewhat later, the cardiac lineage cells or cardiac stem cell were tried. Recently, a sort of mix of different kinds of stem cell is highlighted. Among these various sources of stem cell, I and Professor An's lab focus on the mesenchymal stem cell or mesenchymal stem cell-derived cardiac lineage cell for cell therapy. MSC cell have many advantages shown here. They are quite easy to collect, even we can get MSC from peripheral blood. And it still has multipotent and less likely induce teratoma formation. And mesenchymal stem cell have high compatibility, so it rarely induces graft versus host disease. Based on these advantages, many of clinical trials are still going on with autologous or with allogenic cells. Recently, many of MSC cell trials are going with cardiac commitment cell, but most of the step requires a long time. In the previous study, An's lab dramatically reduced the priming time as short as 1 day. If we properly set up this protocol, AMI patient administered in the emergency room can have autologous MSC delivery before discharge. It would be a very great ideal for patients. Okay, let me briefly summarize the previous results. When overnight epsidin treated MSC cells were injected to the myocardial MI area, the ejection fraction was improved and the fibrotic area was reduced. During this event, YAB1 has gone abruptly, and the knockdown of YAB1 itself induced cardiac commitment of MSC cell. With combination of two kinds of stem cell, YAP high cell and LAB low cell, and this effect were more improved. During overnight treatment of epsidin, airborne was decreased in MSC cell, and the multiple cardiac marker gene were induced, GATA4 and KS2.5 and cardiac 3.9. So we fused the YAB1 with EGFP, and we treated epsidin in the cell. So as you can see here, even short term of the treatment of epsidin, many of the YAB1 already shuttled into the nucleus like this. So after 24 hours, many of the YAB decreased in the total cell. Only few YAB1 level are detected in the only nucleus. Okay, so we question, in this study, we questioned how does YAB1 shuttle into a nucleus and which molecule does determine the fate of nucleus YAB1. We already showed overnight treatment of hepcidin finally results in transcription arrest, but decrease of YAB1 protein level is somewhat faster than inhibition effect. So we tested whether inhibition of protein level are underwent the different pathway. So we inhibit the de novo synthesis by cycloheximide and echinomycin B. As you can see here, the YAB1 still remain enough in the same time treatment of hepcidin. Yeah. So we thought active degradation mechanism. Cloquinine failed to restore the YAB1 level. However, the MD132 successfully reversed YAB1 protein level. So we think that both transcription arrest and the proteasomal degradation are involved in acute ablation of YAB1 in hepcidin treatment. But we think the proteasomal degradation looks more important mechanism. Next, we tested in which subcellular localization of YAB1 underwent degradation. As you see, hepcidin deplete YAB1 in the cell, but when MD132 was treated, the most YAB1 detected in the nucleus. And because MD132 recovered both cytosol, and the nucleus YAB1, so we treated the nuclear export inhibitor, neomycin D. So nuclear YAB1 was not increased here, yeah. So it means cytosol 2 nucleus shuttling is main mechanism of the YAB regulation. As shown previously described, post-translational modification of YAB1 is well studied, especially the phosphorylation of the YAB1 and subcellular restriction is well established. YAB1 phosphorylation, specifically serine 127, and further multiple phosphorylation and recognition at 1433 protein, it restricts the YAB1 in the cytoplasm. PP2A phosphatase remove YAB1, and then YAB shuttles into the nucleus. So we tested our mechanism also follows the phosphorylation dependent signaling. We utilize the commercial available phosphatase to maximize the phosphorylation status. As you can see here, that we found nuclear YAB1 still supershift band, as you can see here. And we treated the phosphospecific 127 YAB1 antibody, and this is I'm sorry. These bands are the positive to the phosphorylation. So we also tested the hepcidin-dependent nuclear shuttling, also same pathway. So we fractionated the cytoplasm and the nucleus and performed the same experiment like this. As you can see here, you can see very increase the size of the bands here, and also characteristics of these bands are positive to the serine 127 phosphorylation. Oh, then we said we should question phosphorylation independent shuttling mechanism. So we simply questioned hepcidin is a HTAG inhibitor, so we tested hepcidin also affect its acetylation level of in the cell. So we found overnight treatment of hepcidin tubulin also slightly acetylated. So tubulin acetylation is regulated by HDAC6. So we also tested the tuvastatin, very specific HDAC6 inhibitor. And as you can see here, the tuvastatin also phenocopy of the epsidin treatment. Yeah, so treatment epsidin successfully induced the acetylation of YABON, as you can see here. And then you can see the dissociation from the protein 33 molecule, cytoplasmic restriction molecule. So we very shortly treated the YABON, very short, about less than 2 hours to minimize the degradation and the shuttling. As you can see here, the epsidin already induced the acetylation of YABON in the cytoplasm. So we consider of the cytoplasmic HDAC, which is subtypes regulate YABON acetylation. As I told you, the tuvastatin is very specific HDAC6 inhibitor. So we knock down the cytoplasmic HDAC6 and HDAC10. So you can see here that we can see the decreased level of YAB1 after treatment of siRNA of HDAC6 and HDAC10 or both. And YAB1 level is decreased, and also neutral targeting, as you can see here, also by the HDAC6 and HDAC10 shRNA treatment. So far, we show epsidin inhibit the cytoplasm HDAC6 and HDAC10 and they regulate YABON acetylation. This induces YABON nuclear shuttling. Then, each molecule degrades nuclear YAB1. MG132 successfully reversed epsidin treatment degradation, as you can see here. This means YABON degradation underwent proteasomal degradation. We tried more our best to get the evidence of polyubiquitination with several kinds of commercial well-established polyubiquitination assay, but we failed. So there are two options in this kind of situations. So one is pushing student more until getting desired result. The other option is finding another possible mechanism. So we searched polyubiquitination independent proteasomal pathway. We searched the different nuclear proteasome machinery, and we found PA200, also named PSME4, is a member of atypical subunit of nuclear 20S proteasome. And also we found on reference PSME4 specifically recognized acetylhistone and degrade the acetylhistone without any polyubiquitination. So we tested the PSME4, so we generated both clone system to control the expression of PSME4 and also generated the constitutive system and tested the YABON level. As you can see here, the YABON level is still remained after treatment of epsidin with a doxycycline. Also, YABON dramatically induced and accumulated in the nucleus with the constitutive virus. We generate both we tested the cardiac commitment after deletion of PSME4. As you can see here, epsidin was not able to fully induce cardiac commitment when PSME4 virus are infected. We also tested a transient transfection of YAB1, and it also shows a similar effect of PSME4 knockdown. So in short, abundance of YAB1 in the nucleus inhibit the cardiac commitment. We bought PSMA4 null mice from Lichen, Japan, and confirmed the deletion of PSMA4 and also confirmed high level of PSM YAP1. And we isolated the bone marrow and primary culture induced an MSC cell and tried to inject MSC cell directly inject in MI induced heart, but very disappointingly, PSMA4 null mice showed even aggravate myocardial death or sudden cardiac death. We are in the middle of evaluation why PSMA null accelerate death. Anyway, the deterioration after that direct injection of PSMA4 knockout MSC cell in MI-induced mice heart. So we returned to the ketone system. We infected these two different virus in the MSC cell and delivered to the impact area and treatment of doxycycline with drinking water. As you can see here, when the PSMA4 deleted by the doxycycline, the scar area is not reduced, and we checked the ejection fraction after treatment of doxycycline, very slightly improved by the MSC treatment. When you see the heart failure level, I'm not sure that we also can say that 50% is a margin of the mouse heart failure threshold. However, anyway, the decreased ejection fraction was more frequently observed in SHPSMD4 treatment group. So this is my summary. Hepcidin induced both transcription arrest and YAP degradation, and also induced acetylation of YAP in the cytoplasm by inhibition of ACTX6 or ACTX10, and then acetylation of YAP induced cytoplasmic nucleus. PSMA4 are the degraded acetyl YAP1, and you have to remove YAP1 to induce cardiac commitment. So we have to figure out some more things like this. Why the YAP1 should go into the nucleus, and which residue are acetylation target, and which acetyl transferase regulate this mechanism, and how about TAS? We have to solve these kinds of questions. I'd like to thanks to my lab members, Somi Yoon and Mira Kim, and I also like to thanks to Professor An and Yongsoo Kim, and also thanks to the Rajan Jain of current PI. Thanks for your attention.
Thank you, Professor Professor Young-Gun An is went out right at the moment for other urgent business, but he's gonna come back soon. So I'll introduce panels briefly before our discussion time. Dr. Hyung-Seok Kim is the professor of Department of Forensic Medicine in Cheonam National University, and Dr. James Jang from Stanford University. Nice to meet you. And Dr. Dong-Im Cho is Cheonam National University Hospital. And we'll have some Q&A and discussion time for 15 minutes. And you can share your comments and Q&A to each other. And then we will have some refresh time for 15 minutes. Please. Who's gonna start?
Maybe I can initiate. First of all, thank you for a wonderful presentation for both Dr. Liao and Dr. Wu and Dr. Om. I think I have a general question to Dr. Liao and Dr. Wu. So your model looking at the neonatal regeneration, and also I think Dr. Wu's model partially reflect those in endogenous proliferative pathway that occurs in the kind of cardiomyocyte. I'm wondering, what do you think the impact of the metabolism in this, cardiac regeneration kind of process. Because from my memory, I think when cardiomyocyte commit or divide, they tend to be more like glycolytic versus more mature or the other cardiomyocyte. They're likely to be more the oxidative, metabolic kind of pathway. So I'm wondering, in your model, what do you think the impact of the internal metabolism in the proliferation of the cardiomyocytes?
So was that question for Ron Lee
Yes.
to answer? Okay, Ron Lee, you go first.
So because we're looking at that, it's actually the inhibition of the well-known neonatal capacity still remain the regenerations. So it's still a baby step, because I think we know at that stage they, consistent with regeneration capacity, but adults' hearts, which Sean is studying, pretty much at lost. And then, with other ways saying, right now using potentially iPS-derived cardiomyocytes, for the cell implantations, or try to find a way. My personal preference, if I may, it's probably find the tune of its endogenous regeneration capacity and to stimulate that. And, the thing got lost may be different with a different disease. So in this case, we're still looking at our molecular, because we just sent out sequencing. So we have not really get back to all the data we needed to looking at what exactly the smoking inhibit the regeneration, what button they actually pushed. So that's, we hopefully once we understand more of those, then we can try to counteract with whatever the signaling pathways or molecule got inhibited to reactivate that, and with certain transient time. But philosophically, I believe adult hearts has a limited regeneration capacity for a reason. Because for organ constantly need to contract and relax, you cannot be always have those new myocyte form, because once it's immature myocyte, you have to be from immature to be mature, right, over time. So then maybe actually they will generate potentially arrhythmias or other impact. So I think, regeneration still the holy grail medicines, but how to go about that, everybody has their, kind of own thoughts and a recipe. I think this really take a whole community, many community trying to figure out. And also, Sean, they study using iPS actually goes through the precision medicine, right? So because you got the different person's iPS cell, right? That's how you study this person's, things. So that's my take. We still distance away from truly regeneration.
Yeah, so I agree with what Ron Lee says. I think these are all very important consideration when it comes to trying to do adult heart regenerating. If I were to get back to the metabolism question, which if I understand correctly, is that you're wondering with these process of proliferation, which tends to couple with a more glycolytic process. So, what are the ways in which metabolism might be regulated or need to be regulated if you want to achieve a regenerative process? If that's what I'm understanding correctly, on what the question, James.
Yes, that's correct.
Yeah, okay, so I think there is clearly very well described data supporting the process of maturing is accompanied by the switch from glycolytic to a fatty acid oxidative metabolism. So that I think everybody's very comfortable with. There's a little bit of data to suggest if you modulate the oxidative environment and potentially the metabolic substrate used with that modulation, you could be enhancing and achieving more proliferation. So there's a handful of papers, especially Hasham Sadak and others have been looking at that particular strategy. But whether or not all of these things, which had all been studied in the fetal or neonatal heart, can actually achieve the degree of adult regeneration. Mostly, I don't think we really know very well, because I think everybody recognizes that once you get to the point to be an adult heart, it has now changed so much of all the physiology and the expected functional requirements that if you were to start unwiring some of these things, what would be the consequence? So the toxicity element Ronli mentioned, I think, is particularly important. And as of yet, I don't think we know how to regionally or specifically perturb just a few set of cells, say right around the infarct region, for example, that you can then stimulate just the ones that you want next to the infarct scar to get that to expand the way that the zebrafish heart, as I was showing in that zebrafish study from Didier Stanier. So the zebrafish somehow figured it out how to do this de-differentiating, sarcomere disassembly, reversing metabolism, proliferate cells, and migrate and repair. And we as humans and most mammals completely lost that. So, I don't know metabolism alone is going to recover everything, but I do think it's going to be sort of one component of this process.
Thank you.
Yeah.
So,
And
I would like to ask Dr. Shanu.
Yes.
Thank you. Thank you for your great lecture. You talk about massive expansion of iPSC-derived cardiomyocyte, right? And what are the difference and advantage of your culture method compared to another large-scale culture method? So, example, the 2D or the 3D, the aggregate culture method. What difference and advantage?
Yeah, so I'm very glad you asked this question, because it's a fantastic question for how do you achieve the kinds of large scale on the practical level. Because one of the issue that I think everybody recognizes is that it's very difficult to have to continuously passage and repassage and repassage, because that's time intensive to do in the 2D system. And if you wanted to create engineered tissue, which is a 3D system, you would not want to have to disrupt the 3D, and you like to continuously promote the expansion in the 3D format. So what would be the ways that might be allow for you to do that? So that's one question which I mentioned. So Lee, my senior postdoc, is actively investigating in a 3D tissue format, what are the inhibitor equivalent of cell contact that you can implement so that the cells can grow in that 3D shape without sensing the 3D environment? So they basically behave like what it grows by itself, sparse, yet they are in a 3D format, so that eventually, after all the proliferation has taken place, so you grow the tissue as a whole 3D tissue, you can then stop that process and get that final fully developed tissue like what you would normally see in vivo. So, we are actively trying to find all these different regulatory signal to be able to grow that as a 3D, recognizing that the cell contact is a real, phenomenon that, that's inhibiting that process of proliferation. But, we don't have an answer, but it's a great question. Thank you.
Thank you.
Okay, so maybe I think it's my turn. Yeah, so thank you for some great lectures, three professors. So I have two questions. The first one is to Dr. Liao for general question. And the second question is to Dr. Wu, Shan-Hu. So first, my question to Liao is the glucose level in your experiment condition, because some nicotine and acute stress is some major risk factors in cardiovascular disease. So we know that these conditions maybe increase the glucose level. So I want to know some glucose level changes in your experiment models after nicotine consumption.
So
So second, yeah.
Sorry, maybe one at a time, so I won't forget what's the question. So for the glucose level, that's, it's actually very important questions, but unfortunately we did not measure that. And we don't believe, because it's a neonatal heart, we did not do any intervention besides smoking. So now you are asking if smoking, if I got that right, it's potentially we're changing the homeostasis of that. So we are looking, actually, we just sent out some sample to looking at the metabolomic. So we're trying to using the angle to looking at the global, the metabolomics, changes, and then from there maybe come back to it. And I, early on, talking about metabolism, looks like this group has a lot of interest on the metabolic regulation. Personally, I feel that's really a very important parameter. Just something very, the example of how metabolism can affect us is actually the quickest way to lose weight actually is control your diet, right? Control your metabolism, then you can really change it, right? So quickly. So that's actually, something all associate and come with every living system. And I think we indulging ourselves into chocolates, sweets, and something, and not eating vegetable and not the balance. So we also perturb our, metabolism. So I think that's a very, very critical things, and we are looking into it. So hopefully can have answer sometime later.
Okay. So my second question, moving to Dr. Wu. So my interest in cardiac stem cell and some stem cell-like cells is LGR5 function. So as we know, some LGR5 is very important to preserve the stem cell characteristics. So my question is, you using some YAP inhibitor. So is there any effect to sarcomere function through some LGR5 pathway?
Yeah, so a great question. So we do not look at the LGR5 largely because that's not a gene that really is described much in the cardiac biology. There was one paper from Ken Chen's group that looks at an outflow tract region that in the early fetal heart that may have some LGR gene expression, but subsequently when the cardiomyocyte differentiate, they no longer have this LGR5 gene. So when the YAP inhibition that we study was intended to look at the role that YAP was described in cardiomyocyte and stimulating expansion of cardiomyocyte. And what was somewhat surprising that I was showing is instead of inhibiting, causing a reduced proliferation, which is what we expect because YAP is known to stimulate proliferation of cardiomyocyte, we actually got the opposite finding, which is that inhibiting the YAP actually stimulated the proliferation. And then so what we realized, YAP is a very complicated molecule, which I think, you heard very well, also from Professor Kwan, that the YAP signal plays a role in many different biological contexts in many different kinds of cells. In our hand, the inhibitor, verteprofen, was actually causing a completely other effects related to probably the cytoskeletal arrangement and cell-cell contact, which is a very well-known role for YAP. And what happened is that inhibiting the YAP in that situation made the cells change into the spindle and disrupted the sarcomere structure. So in our case, we saw the sarcomere structure disruption in the treatment, to inhibit the YAP. And that actually led to this completely new direction and idea about how sarcomere assembly is actually a factor that inhibits the cells from proliferating, which I think people sort of wonder about and didn't really have good data to say for sure. And I think our data plus the paper from Didier Stanier that I show in zebrafish clearly establishes that the sarcomere organization needs to be removed and disrupted if you want the cells to go through the cycle activity.
So,
okay, thank you.
Okay, go ahead. Professor
Can I give a simple question to Professor Yeah. Taz is a very famous partner of YAP1, right? So are you gonna doing some experimental doing on for the finding the effect of epsidin on Taz in mesenchymal stem cells?
We still didn't check the TAS in epsilon treatment or some differentiation condition, but we found very unchanged level, even level in any different conditions. So usually TAS localized in the nucleus in mesenchymal stem cells. So we didn't check the interaction after the shuttle into the nucleus of YAV and TAS interaction. We didn't check about that, but in the later we will check about the kind of mechanism. Thank you.
Okay, so I think it's maybe I'm very sorry, Dr. Long, because I had to do something for the hospital. So, but I have rejoined with this meeting. I just one brief question for the Sean. I think you was some Wnt beta-catenin signaling activation, and then some reduced cell contact probably is very wonderful situation for the cardiomyocyte proliferation. But still, I wonder that because if we think about the human heart, it's a very huge size. Even though we make a very, very the active proliferation of cardiomyocyte, can you really believe that this kind of strategy to replace in the damaged heart, especially in the field of some myocardial infarction or something? So we really need to make the huge proliferation of cardiomyocyte, or can we make stimulate the host dependent mechanism to making the healing process for the cardiomyocyte proliferation? Or I'm not sure that so how can we make the future?
So you asked a very important and very insightful question, and that is, if we wanted to do treatment, can the beta-catenin Wnt signal pathway be leveraged for the purpose of the regenerating activity from the endogenous heart?
Yes.
And I think that is a more challenging situation because the Wnt signal in general participates in many different biological processes. So beyond what we show for the cardiomyocyte, there's also activity of Wnt on fibroblasts, epicardial cells. So to do the therapy overall is going to be challenging because you also now perturb other processes. But so what the mechanism that we identify is something, at least in our sort of concept, is going to involve much more of engineering tissue separately as a ex vivo created tissue, and then looking for if you can create this large size ex vivo created tissue and then try to find a strategy to re-implant and reintroduce into the infarcted region that gives you a much more of a integrated and organized repair in one large regions, rather than trying to identify the local endogenous cardiomyocytes and find a way to get it to de-differentiate and then stimulate the proliferate and then forward differentiate, and trying to do it in some way that doesn't disrupt other cell type, which I agree with you that it sounds like a very difficult strategy. At least not going to be one strategy that's going to do all of those multiple step process.
Okay, Dr. Sean, thanks. So I think it's maybe we have to close in this session first, and then we will meet again in the session second. So please take a time for the breaking, right? Young-soo Kim, right? So
yeah, right.
Okay, thank you. So see you soon.
Okay. Actually, as I briefly said in the break time, this is Stanford Korea symposium was my dream symposium. So I'm really honored to be here and to present my postdoc work. I always show the embryos. This is mouse embryos from early embryonic day 8.5 to 17.5, because in my PhD program, I studied the liver and pancreas development, which is endodome organogenesis from these stages. And in 2015, I moved to the Stanford as a postdoc in Chris Redor's lab to study cardiovascular development, since I recognize endothelial cell is important for the organ development. As you see, the coronary vessels supply the blood to support the proper heart muscle function. So if you lose or block the artery or vessel, then the cardiomyocyte die, or that's affect the human life immediately. Our lab, or Chris Redor's lab, we study cardiovascular biology, especially we are interested in the multiple progenitor cell, endothelial cell progenitor cells, how those progenitor cells differentiate coronary vessel endothelial cell, and also the artery. So our lab has multiple research interests, including heart development and congenital heart disease modeling, or stem cell differentiation and heart regeneration. Among these, I'm very interested in the number 1 to 3, heart organogenesis and the congenital heart disease and stem cell differentiation. But as the Longley previously briefly explained, we realized the neonatal heart regenerate. So also the Christie really move on to her interest to the heart regeneration. So almost 70–80% the lab members working on the heart regeneration project. So before I introduce my project, I always ask people where or when the first coronary vessel formed, because we should know. To understand how the coronary vessel regenerate, or fix the a sick vessel, we should know how the coronary vessel develop. So interestingly, this is the multiple people, including Christie or other the researchers found. There are 2 progenitor population, but their location and the time point is different. So first, the area, the one progenitor cell contribute to the coronary vessel is sinus venosus. That is located at the backside of the embryonic heart. So that area, the sinus venosus, differentiate to the endothelial cell. As you see the confocal microscope with the VCAT hearing, you can see the first the vessel form in the backside. Little later, the other endothelial cell type, which is endocardium, which is endothelial cell, but the not sprouting form, but that is form already embryonic day 8.5 and 9.5, but that is not the angiogenic endothelial cell. But at around embryonic day 12.5, that endocardium start to invade into the myocardial wall, ventricle wall. As you see, this is the ventral view. So the another set, the endothelial cells sprout the inside to outside in the ventral way. Those 2 multiple progenitor cell contribute to all the capillary cell, or coronary vessel. Those vessels are also differentiate into the artery once they got attached to flow, with the aorta, and also those cell types differentiate further coronary artery, vein, or capillary. When I joined our lab, I recognized most of people are interested in the endothelial cell differentiation. To differentiate my research from the other lab member, I thought I was looking for the other project. I found the interesting like observation, and also I brought the hypothesis. What's the role of the coronary vessel during the heart development? So my hypothesis on the coronary vessel may important role in the heart wall thickness. The reason I made that hypothesis, as you see the section image of the early embryonic heart from 9.5 to 15.5, every 3 days, you can see the change of the cardiac wall thickness. So approximately 10 to 20 micrometer at 9.5, you can see after 3 days, it will double. But the interesting after 12.5 and between like these 12 and 15.5, you can see dramatically the cardiac wall thickness is increased. So there are important biological events in here, like a trabeculation, and you can see like a finger-like muscle cell formation, and other one is compaction process. So as I just briefly explained, I can show the other SEM image. You can see until 12.5, the thickness is not that much different, but after during between 12 and 14.5, you can see significant difference. So as I just introduced, there are two distinct populations, multi-progenitor cell. One is from inside, the other is outside, like a sinus venosus or endocardium. So my hypothesis, this events like invading or like a two progenitor cell contribution may be important for the thickness formation, cardiac thickness formation, which is important for a proper cardiac development. But to test my hypothesis, I need the animal model. So I was working in the biology department. So I was able to collaborate with the faculty member in next lab, who is the Ashby Morrison. So she was working on like a chromatin remodeler INO80. And what is that INO80 is, which is like a like a protein, but that also they work on as a like transcription factor. They recruit the other protein molecule, and they do like lots of different function, including stem cell pluripotency. The INO80 play important for the stem cell pluripotency or cancer or metabolic states. But actually the Christian, the Ashby thought maybe the INO80 also important for the cardiac function. So my first project was what's the role for INO80 during the heart development. So let me see. To know the INO80 role during the cardiac development, we generated multiple transgenic mouse line using conditional knockout system. So we knock out INO80. Firstly, the reason we chose INO80, the INO80 knockout mice that die before gastrulation. So we need to use the conditional knockout system. So we knock out INO80 in the cardiomyocyte and fibroblast. We didn't see any phenotype. But when we knock out INO80 gene in the endothelial cell using type 2 Cre, and we see the dramatic phenotype in the heart. especially the heart wall is really thin. So it was confirmed by other marker, and the mucin is also the label in the endocardium. You can see the outer wall, like a compact myocardium zone, and trabecular myocardium zone is like almost the break, like a really became really thin. So this is not, but interestingly, this phenotype called like non-compaction phenotype, but this is not only for mouse. This is happened in the human as well. So, but interesting part, as you remember, I just explained we knock out INO80 gene in the endothelial cell, and phenotype occur in adjacent cell, which is cardiomyocyte. So this could be just secondary effect. So okay. So when I study or define this phenotype, I actually visited like a Joseph Wu lab and talked to clinicians. And I explained the phenotype, and I found the LVNC phenotype when we knock out some specific strong or important gene in the endothelial cell. And couple of people didn't trust or was not interested because LVNC is known the human disease, but the most people understood the LVNC caused by the mutation in the cardiomyocyte, not endothelial cell. So, briefly, LVNC is a left ventricle non-compaction. Basically, the left ventricle is much thicker than right ventricle, but this is kind of the third most popular cardiomyopathy, and also it is known more found in the infant, or 13% mortality rate for children, but there is only a treatment that is the heart transplantation, but still it's very hard because it's a congenital heart disease. So we need some proper animal model. So I thought this endothelial cell derived LVNC mouse model can be a novel model for congenital heart disease. So briefly, what's the phenotype of this mouse model? Briefly, when the vasculature is disrupted, the proliferation in compact myocardium is really reduced, as you see here. And the other phenotype is, interestingly, no molecular marker were not changed, but the morphologically, we can see it's an intermediate area. The cardiomyocyte start to they make a more sarcomeric shape. So they lose proliferation and they increase like a sarcomere, or it's hard to say the maturation, but we see the more sarcomere structure in the area. So I more say looks more mature because the difference between the CM or compact and trabecular myocardium. So I thought, we proposed this model to angiogenesis drive ventricle non-compaction. We confirmed this model with a different Cree line, so that's I'm not explaining, which is published in the 2018 paper. And also I just briefly introduced, we knock out the INO80 in the endothelial cell, but the cardiomyocyte affected. But to understand that, this system, I had to some in vitro system to prove or to test more mechanism. So since we don't have proper organoid system or a co-culture system to mimic our phenotype, so I set up I set up this novel embryonic explant system. So simply, I take out the mouse embryo or 12.5, then culture it. In this stage, after I culture this, and you can see the endothelial cells sprout, but the CT&T cardiomyocyte expand accordingly. As you see, the mutant, they fail to form proper, or the endothelial cell fail to migrate, and the cardiomyocyte also affected. So which support our idea, angiogenesis important for the cardiomyocyte growth or expansion. But still we don't know what is the factor regulate this cardiomyocyte. So even though we published it, that the result by just showing our the angiogenesis drive LBNC mouse model, reviewers ask, or other people ask, okay, we kind of trust your idea or your result. Then what is the secreting, possibly the a factor that regulate cardiomyocyte, or some factor may affect the cardiomyocyte maturation or the proliferation. So while I was doing the my project, Chris Redo's lab really focus on the single cell analysis and publish several great papers using single cell RNA sequencing data. So I proposed my idea: single cell analysis, single cell sequencing analysis can make me find some angiogenesis drive or paracrine factors that influence cardiomyocyte proliferation and maturation. So I'm going to briefly show how I identify some molecules and test those angiocrine factors which affect the cardiomyocyte characteristic. So my hypothesis, I use the control and type 2 CRE-INO80 because this is a very strong phenotype. So I was lucky that I had this model. So I ran, even though we had the bulk RNA-seq, we didn't find any clear molecule. So that is why the single cell RNA sequencing is powerful than just bulk RNA-seq. That's what I'm going to show. So I run the single cell RNA sequencing to find the molecule that regulate cardiomyocyte proliferation and maturation, and I validate the candidate in vitro whether the candidate molecule mimic my hypothesis. Briefly again, as a developmental biologist, this the morphology is super important. So you can see the endocardium, the endothelial cell always support trabeculae, and they are more proliferate cardiomyocyte located in compact myocardium area, but the coronary vessel endothelial cell located here. So my hypothesis, factors from this population will increase proliferation cardiomyocyte. This endothelial cell or the defect, some factors from some defective endothelial cell will increase maturation or something like I made this hypothesis. Then to isolate that some secreting factor. So as I said, I run the 10x sequencing using the 3 normal heart and 3 angiogenesis drive LVNC heart, and we were able to subcluster 9 different populations, then including endocardium and coronary vessel endothelial cell, which is clearly 2 distinct population. So we subclustered the endocardium or endothelial cell, which is include the 2 endocardial and mutant-like population and coronary vessel endothelial cell. Interesting, you can see when we split this single cell data into control and mutant, and we were able to see some intermediate population. So one of powerful things in the single cell RNA sequencing, we can identify some disease-related population. So I thought this could be some important cell population in which cause our phenotype. So just briefly, like using the known marker of the endocardial and coronary vessel, and we were distinguished this is endocardium and coronary vessel endothelial cell. And previously we showed the INO80 knockout endothelial cell showed the like a hyper-proliferative property. So interestingly, the endocardium is mostly not proliferative, but in our INO80 mutant, even though they are labeled endocardium marker positive, they are more proliferative. So we were able to see this is mutant 1, and we validate with the other markers, and by section and EDU, like a S-based marker, like a proliferative marker, prove the defective endothelial cell located like a non-compaction area, so which were quantified here. Then I try to isolate some factor which, as I explained, like induced proliferation and other factors which which cause the LVNC phenotype. So what I did is I list up all the like 167 the like genes or factors, and I excluded based on my category or standard. But it should be expressed specifically coronary vessel endothelial cell, and also affected by loss of INO80. But interestingly, this collagen 58-1 was the new kind of marker, which is expressed more coronary vessel endothelial cell here, which is located here, compact myocardium area. It is also the affected by loss of INO80 specifically. As you can see, the coronary vessel endothelial cell here, knockout, collagen 58-1 was completely gone, and in the single cell data, also in situ. So I want to test, okay, then collagen 58-1 could increase the cardiomyocyte proliferation. How? First of all, I mean, I established very nice the explant culture, as I explained. So I use that one. So when I treat the collagen 58-1, which is a recombinant protein, into the normal the explant culture, it is this is explant culture, so it includes coronary vessel and cardiomyocyte and fibroblasts to really mimic the in vivo. But when I treat the collagen 58-1, then it increased proliferation. And the best thing of this explant culture, we can do rescue experiment, ex vivo condition. So I have shown INO80 knockout one. This is the good thing is cardiomyocyte is normal, like a wild type, but they lose proliferation. But when we treat the collagen 58-1, and we were able to rescue the phenotype. And also I was mimicking that phenotype by adding a VEGF receptor inhibitor. So we almost removed the vessel, and also that's like a cause reduce cardiomyocyte proliferation, and adding the collagen 58-1 was able to rescue the phenotype partially. And not only that, by collaborating with David and Joseph Wu, and we were able to test collagen 58-1 can induce proliferation in human iPS cardiomyocyte. David and the co-worker Johnson provide the human iPS cardiomyocyte, we established the most similar the status of the cell, and the adding collagen 51 into the wild type also increased, but we were able to use the LVNC human iPS cell cardiomyocyte, which is published in the 2016 in Joseph Wu lab, and we use the human patient, the cardiomyocyte, which were rescued by adding the collagen 51. So it was the experiment we performed. And the other story, like I'll do a little faster. And I was selecting the other molecule, which is increase in the mutant area. I saw more the factors, which is increase endocardium. So as you see here, then I confirm with the other filing plot. So I always validate by the in situ hybridization, so RNA level, and you can see the TGA-beta induced TGA-I is significantly increased in the non-compaction area. And for example, TGA-beta-I or IGBP3 was increased in time as time goes, like 12.5. And so I chose a 4-factor. And interestingly, I compare with the 13.5 and 14.5 Burkhardt-seq, and this is we run the Burkhardt-seq from the HUVEC cell, and we think the TGI-beta-I and IGBP ISG615 was affected by INO80 directly, and IGBP3 and ADM could be just affected by the hypoxia somehow. So what I did is I treat the dose factor, which is increasing the mutant, to see whether those I can mimic the non-compaction phenotype. One is reduce proliferation. Interestingly, I see we saw the 4-factor reduced clearly, significantly reduced proliferation, and individual treatment also mimicking same phenotype. But interestingly, we didn't see the synergistic effect. And not only reduced proliferation, we saw the cardiomyocyte also start to express more cytometic features, and also structurally elongated, which kind of mimicking the LVNC phenotype. Also, we tested whether this 4-factor or individual factor increase or decrease the cardiomyocyte proliferation, and also the cardiomyocyte structures. And by collaborating with the David and other Najat, and we were able to see the treating this factor or mimicking some more maturation characteristics. So, and this is another one. I know people asked whether knockout INO80 affect or mimicking LVNC phenotype in adult states, but as you see, the titer Cree INO80 knockout, the mutant die while they're birth, like less neonatal mice. So we were testing that hypothesis using the emphatic Cree one, which is only we knock out INO80 on one population of the endothelial cell, but that mutant has also some the dilated the cardiomyopathy and the functionally defective. So in the summary, so during my postdoc period, I set up the new LVNC model, like angiogenesis derived congenital heart disease model, and to understand and find some molecule to regulate cardiomyocyte, and we use single cell RNA sequencing, and we introduced some molecule to control the cardiomyocyte characteristic. So I'd like to thank all the Redwood's lab and Crystal lab. This is the last. We always go to some lab retreat, but no longer we can go. But this is the last picture we went to the other place, and actually especially the Crystal Redwood's lab, Christy and Joseph Wu and Ashley Morrison, who are the corresponding author for my paper currently under review, and especially the David. We were working on this project for almost 4 years together by in the Stanford K-BioX. So if you have any, this is my presentation. So if you have any question, I'm happy to.
Thank you very much, Xiang. This is wonderful. It's a lot of work being done. So we're going to ask him a question in the question section. So we're going to thank you. This is really tour de force. So it's wonderful. Enjoy it. So we're going to introduce the next speaker. It's Dr. Yang-Soo Kim, and she's going to share with us on the phenotypic plasticity of cell in cardiovascular disease. Yang-Soo, take it away.
And thank you for all of you for joining us today. And the topic of my talk is phenotypic plasticity of a cell in cardiovascular disease. And the speakers in this symposium are giving us insightful and brilliant stories, and I really appreciate it. And it's probably not enough, but I'm going to show you some preliminary data of ongoing studies. Next. I will skip the boring background. So next, further 2 slides. Next. Yeah. The concept of cellular plasticity was first introduced by Dr. Helen Blau in 1985 to denote the capacity of a cell to change its identity, and she's working in Stanford now. The grooves indicate the differentiation state, and the pluripotent stem cells have potential to roam down these several groups. Plasticity is a common use term in dedifferentiation, transdifferentiation, and now we are having unsolved questions whether tissue injury can change the threshold for those groups. The specific definition of plasticity that I use for this study is the capacity of cells to change their function and phenotype in response to environmental cues. Next, next slide. Yeah, there are several types of dynamic cell transitions such as EMT, MET, and endo-MT. And now I'm going to show you a sort of preliminary result with 2 parts: myocardial infarction and atherosclerosis model. Next. This is a human autopsy sample showing a cardiac region which is composed of dead cells, fibroblasts, and immune cells such as macrophages, sometimes neutrophils. In our laboratory, we've been trying to emulate pathologic features of human disease heart for the further translation research. Next, please. Over the years, we have experienced successful therapeutic applications of small molecules, adult stem cells, and growth factors. Next. To find out the underlying mechanism in cardiac disease, we started to screen NF-kappa B pathway, a notorious regulator in inflammation, tumorigenesis, and degenerative pathologies. Luckily, we have found that IKK epsilon knockdown macrophages showed higher INOS induction, suggesting the involvement of IKK epsilon in inflammation response. INOS is a representative inflammatory marker, and IKK epsilon is an inducible kinase classified as a member of non-canonical NF-kappa B pathway. Next, and next. To convince ourselves, we first compared the pathologic progress in a myocardial infarction model in both in wild type and IKK epsilon knockout mice, and found that cardiac function and fibrosis worsen in the IKK epsilon knockout group. Next. To determine the potential mediators of higher inflammation in IKK knockout macrophages, we arrayed phosphorylated proteins, and phospho-P38 was one of the candidates. And we verified the downregulation of phospho-P38 and higher induction of INOS in IKK deficient macrophages. Next. And one more click. Yeah. To recapitulate the IKK knockout macrophages, we used a P38 inhibitor as the compound, and as the treatment increased INOS induction in macrophages. Next. Then we isolated the cardiac macrophages from the mouse heart, and you can see the phospho-ATF2, a well-known substrate of P38, was lower in the IKK knockout cardiac macrophages. Next. Then we wondered the association of P38 and cardiac injury. Click twice. And we found that the phospho-P38 was significantly decreased throughout the MI after 1 day in a mouse model. And in IKK knockout mice, phospho-P38 was markedly downregulated in both ischemic and non-ischemic cardiac injury model. Moreover, human failing hearts also showed the reduced phospho-P38. Next, and click one more. And previously, we have showed the substantial cardiac repair with small molecules such as 5-azacitidine and bio by inflammation responses. And now we realize these two molecules restore the phospho-P38 in stimulated macrophages, and this could in part contribute to attenuate cardiac damage post-MI, I think. Next. Then we examined whether phenotype of cardiac macrophagy was more inflammatory or not in the IKK knockout group. Unexpectedly, however, the number of anti-inflammatory cardiac macrophages was much higher in the knockout group. Next. Then after several sub-analysis, we found that the large portion of anti-inflammatory macrophages also expressed alpha-smooth muscle actin, which is a marker of myofibroblast. I'll click one more. At the beginning, I showed you that cardiac fibrosis was larger in IKK knockout mice. So we suppose that macrophages with low P38 activity might be associated with some kind of cell transition to fibroblast to induce more fibrosis. Next. In cardiovascular disease, I'll click one more. Cell transition and transdifferentiation were already reported, but macrophagy to fibroblast transition in the heart tissue was first reported in 2019. They clearly show the expression of myofibroblast markers in macrophages in the infarcted myocardium. Next. Myofibroblasts are the major collagen producing fiber to cell type during active fibrosis and originate from resident fibroblasts, epithelial cells, and endothelial cells. Crucially, macrophages, especially anti-inflammatory macrophages, can transform into myofibroblasts in a process termed MMP as a novel mechanism for the involvement of macrophages in fibrosis. Next. Again, this graph clearly show that the portion of the inflammatory macrophages were not different in both groups, but larger amount of anti-inflammatory macrophages underwent MMT in the IKK knockout group. Next. So to recapitulate MMT in cell models, we differentiated human monocyte cell line THD1 or human blood-derived CD14 positive monocytes to anti-inflammatory macrophages by treatment with interleukin-4 and interleukin-13, and then stimulated them with pro-fibrotic mediator TGF-beta1. Alpha SMA myofibroblast marker was upregulated over time, indicating successful MMP induction in cultured cells. Next. We showed a lower phospho-P38 level and a larger portion of MMP in anti-inflammatory macrophages in the IKK knockout group. So we examined the effect of P38 activity on MMP. We treated the anti-inflammatory macrophages with vehicle or P38 inhibitor SV compound during MMT induction. Click more. Change in cellular morphology, which is one of the MMT induction, was much greater in SV-treated macrophages. Click one more. Moreover, upper SMA was increased, and click one more. And fibrotic markers such as fibroblast activation protein alpha and BMNT were also increased in SV-treated MMT model. Next. In a human blood-derived MMT cell model, SV treatment apparently increased the alpha SMA expression in CD206 positive anti-inflammatory macrophages. Next, I'll click one more. So active P38 is associated with inflammation resolution and could be a rate limiting factor for MMT in macrophages. Next, I'll click one more. And the second part is about the smooth muscle cells in vascular pathology. Healthy and functional blood vessels include fibrous connective tissue, endothelium, and smooth muscle cell layer. And you can see the atherosclerotic plaque, which may consist of macrophages. And this plaque is surrounded and kept safe by smooth muscle cell in stable fibrous cap. This plaque rupture occurs when this fibrous cap is getting thinner and weaker. Next. By the way, at the time, we were having some therapeutic candidates for cardiovascular disease, and angiopoietin-like 4, ANGPTL4, is one of them. ANGPTL4 is secreted from mesenchymal stem cells under inflammatory stress to induce inflammation resolution, and we recently reported therapeutic effect of ANGPTL4 on a mouse myocardial infarction model. Next. Given the anti-inflammatory activity of ANGPTL4, we examined the effect on atherosclerosis. Fully knockout mice receiving a high-fat diet were injected with 2 microgram ANGPTL4 protein 2 times per week for 7 days, and compared with mice injected with vehicle alone. The entire aorta and aortic root, blood, and bone marrow-derived macrophages were collected for further analysis. As you can see, the plaque size and area necrotic core, the areas of necrotic core were much smaller in ANGPTL4 treated group. And next, the thick fibrous cap in the left panel covering the lipid core of the plaque is composed of bundles of contractile type smooth muscle cells and collagen fibers and few macrophages. The thin fibrous cap on the right panel, this fibrous cap contains many macrophages and lymphocytes, few smooth muscle cells, and less collagen fibers, and all of which may weaken the cap, leaving the plaque vulnerable to rupture. Plaque rupture is known to be inversely correlated with the amount of contractile smooth muscle cell. So we assess the protein level of transgellin, a marker of contractile cell. Next slide. And this ANGPTL4 treatment increased the transgellin positive area in the plaque. And next, and click one more. Mostly importantly, the fibrous cap thickness was greater in the region of ANGPTL4 treated mice, indicating advanced signs of plaque stability. Next. So, and then to assess the effect of ANGPTL4 on smooth muscle cell phenotype, we treated smooth muscle cell with cholesterol for 3 days with or without ANGPTL4. And we can see the red cells, the cholesterol-laden smooth muscle cell divide form cells, which are known to be detected in advanced atherosclerotic plaque, and click. And these form cells, next. These form cells were reduced by ANGPTL4 treatment. And smooth muscle cells isolated from mouse aortas and cholesterol treatment induced the transient loss of alpha smooth muscle actin, which is a marker of smooth muscle cell. Click one more. But ANGPTL4 partially restored the alpha SMA expression in smooth muscle cell, indicating phenotype of smooth muscle cell could be changed in the inflamed lesion. Next. So to make clear, the atherosclerosis was analyzed in aortic root region. We can see the plaque with a cap with a thin smooth muscle cell layer and upper in lower panel, even well along the layer showed co-expression of CD68 macrophage marker in smooth muscle cell, indicating this kind of unstable plaque has few smooth muscle cell or occupied by macrophage-like smooth muscle cells. Next. On the other hand, in the ANGPTL4 treated group, smooth muscle cells covering plaque lesions safely, so we can suggest ANGPTL4 administration helped the ability of smooth muscle cell to contribute to stable cap formation. Next. In vascular pathogenesis, smooth muscle cells were known to initiate expression of macrophage markers, and this phenotype switching is largely dependent on the transcription factors, KLF4. As expected, ANGPTL4 treatment obviously suppressed KLF4 in atherosclerotic aorta and smooth muscle cells. From this result, ANGPTL4 was suggested to inhibit a phenotype modulation of smooth muscle cells in the pathogenesis of atherosclerosis and contribute to maintain on smooth muscle cell rich and macrophage poor fibrous cap area. Next. To further characterize the effect of ANGPTL4 on cardiovascular disease, we measure the circulating level of ANGPTL4 in patients with myocardial infarction. Mean levels and distributions of plasma levels of ANGPTL4 were shown here, and patterns and patients in the higher ANGPTL4 had a lower rate of cumulative incidence, indicating the possible positive relationship between ANGPTL4 and plaque stability. Next. Stefano De Miller, an outstanding vascular scientist in Frankfurt University, had visited here Gwangju to join the international symposium, which was organized by Dr. An and Dr. Liao several years ago. We can't believe it happened. Anyway, click one more. Recently, she published an interesting article showing that endothelial cells undergo a transient mesenchymal activation with the metabolic adaptation within the first day after MI to facilitate endothelial cell migration and clonal expansion to regenerate the vascular network. So, cell transition engages in many diseases such as cardiovascular disease, fibrosis, and cancer, and we are trying to reveal the pathophysiological significance of cell transition. In summary, of course, we need more experimental validation to be conclusive, but our data up to this moment show that active P38 induce inflammation resolution and limit the cell transition in macrophages. And in atherosclerosis model, we found that vascular smooth muscle cell underwent cell transition to macrophage-like cells, and therapeutic factor ANGPTL4 might limit this transition to contribute to stabilization of atherosclerotic plaque. So we get to suggest that both cell transition of macrophages and smooth muscle cell can be modulated during the disease progress. Next. And I'd like to thank our lab members for their tremendous effort and Professor Young-Gun An for his full support. Thank you.
Thank you very much. This is wonderful. And this is a perseverance, right? And teamwork make it work. So it's wonderful. Okay, so this two wonderful presentation now is open for discussions. So anyone's, I think, can people free to ask? I think so. If you unmute yourself, you should be able to ask in question.
Sorry if I interrupt. I'd like to kind of ask a question to Young-Suk Kim.
Yeah.
So maybe my memory is kind of incorrect, but I think there's a many GWAS hit for the ANG for your target kind of protein marker, and I'm wondering if you know any if there's any SNPs associated with those genes, and also if there any association study have been done in related to coronary artery disease. Yeah.
Yeah, thank you for the great question. In 3 or 4 years ago, there are several papers reported in NEJM that ANGPTL3, ANGPTL4, and ANGPTL8 were inversely correlated with lipid profile. For example, ANGPTL4 is inhibiting LPL, a kind of lipase. But in our study, it's different type of ANGPTL4 activity and ANGPTL4 level in plasma. They have different roles, I believe, because ANGPTL4 has many types of glycylation on their molecule. So the glycosylation and some kind of mutations are affecting on their activity for sure, but total ANGPTL4 protein level is another different story, I believe. So as you mentioned, the kind of SNP or activity study should be performed in our project. So we are collecting the plasma from various kind of patients with MI. So we are working on that. Thank you.
Thank you.
So when you're looking at the sample from the myocardial infarctions, you have a plan to do metabolomic profiling?
Oh yeah, great question. But actually, as a plan, we want to screen or we want to analyze the exosome, DNA, RNA sequencing, and all kinds of protein and metabolomics. But as you know, it costs too much. So step by step, I will do most of them at all, I believe.
Because since we're talking about metabolomic and metabolism early on, so I think that's actually one of the key factor affects a lot.
Yeah. Actually, ANGPTL4 is a kind of hormone sensitive to nutrition status. So your suggestion is great. Yeah.
Sean, look like you have a question.
I do have a question. I had a question for Sion. I'm always nice. I have a question for Sion, who has this very wonderful and impressive study in looking at this INO80 downstream on this cardiomyocyte proliferation, and this interaction between the endothelial and cardiomyocyte has been definitely a great area that many people explore for development and also for potentially regenerative applications. So the question that I have is, I was a little surprised that the extracellular factor release looks like a handful of ECM molecule, and the ECM molecule generally had been considered something that more sort of structural alignment, functional, and not necessarily as a mitogen growth factor stimulation. And I'm curious to see what you think might be the mechanism to the way that the ECM molecules would stimulate the proliferating activity.
Yes, that's a great question. Actually, we are working on that project for the next one. But as you know, the many integrins are highly expressed in the cardiomyocyte. Like the TGA beta I is affected by the IJ integrin. So that's one possibility. And the reason I chose those 4 factors are they're like the known. They have also confirmed in. They have shown in the human like a very similar disease. So which we chose those factors, but we haven't get into the mechanism yet. So that's kind of the next project we are working on.
Yeah, I mean, it does remind me of the work from Eldad Chazor in Israel with the agrin injection,
Yes.
because the
Yes.
agrin was discovered by ECM comparison between mature
Right.
versus immature, and they thought that the agrin treatments is in a way interacting with, say, the dystrophin complex, you know, to mediate some of this activity, you know, between the extracellular matrix and the you know, cytoskeleton. So I'm sort of wondering, maybe there's a parallel between some of these ECM molecule, you know, that you're looking at with what agrin does.
Right. Actually, that's the reason we the focus on the ECM molecule as well. So that's kind of our hypothesis. So very similar. We saw the very similar the phenotype, like some factors affect the proliferation and the other factors and maturation. So kind of that is a one of model of the design of experiment. Yeah.
Super, great, thank you.
Yeah.
Any other questions from the panel?
Yeah, I have a question for Dr. Xian Li. Dr. Li. Yeah. So I think it's a really impressive study about INO80 and enzyclide collagen 50A1 related INO80. So I wonder, what is the molecular function of collagen 50A1? It means, how does collagen 50A1 stimulate cardiomyocyte proliferations? So I wonder.
So there are like a recent paper. We have I mean, actually with I just explained, we haven't done for the molecular mechanisms. In this project, we want to just make sure the molecules can recapitulate our the model. So we're more focused on phenotypical characterization.
Yeah.
But the collagen 51 knockout mice has a very similar capillary the formation defect and cardiomyocyte defect in the heart and even other organ like a leg or something. So, and also we generated collagen 51 conditional knockout. We knock out the collagen 51 in the endothelial cells specifically, and we partially recapitulate our phenotype as well. So collagen 51, also the ECM molecule. So we are testing that hypothesis, but we don't have like a great model system. So that's why I established the explant culture and using the other human iPS cells. So that's kind of, I'm working on it, treating or using explant culture and in vivo model. So yeah.
Yeah, that's all. Thank you.
Wonderful, thank you.
I just remember, if I could ask one very quick question. So
Only for
yeah,
you.
only for me. Thank you. Hopefully it's very, very straightforward. So a question I have for Young-Suk is that the cell plasticity has been such an actively studied and debated topic. And there is one plasticity that I find very interesting, but I have not been able to get a straight answer, which is that there has been report of a smooth muscle that transitioned to a macrophage foam cell-like phenotype. And I'm just curious, is that something that people have really been robustly reproduced and confirmed by many groups, or is it still a pretty actively debated concept?
Yeah, good question. 2014, there is a report, a really brief report on circulation research. I think they showed smooth muscle cell. They had a mouse with a smooth muscle specific beta gal, so something like that. So by using the genetic and tracing model, they after the atherosclerotic induction, in the atherosclerotic plot, there is a much of a large population of smooth muscle cell derived cells, and they showed the kind of visual evidence that cell co-express the CD68 or F480 macrophage markers, and smooth muscle cell synthetic phenotype markers. So they firstly reported the possible strong involvement of smooth muscle cell transition into macrophage-like cells. And last year, similar phenotype phenomena was reported in a very molecular level. So many scientists believe that macrophages can originate from smooth muscle cell or other cell types. But in the atherosclerotic model, especially in advanced model, there are so many reports in the smooth muscle cell can transform to macrophages like cells, but the reversibility is not clear yet.
Okay, great, thank you.
Okay, I think this is a very lively discussion. I'm sure we have more questions, but to respect everyone's time, because we probably a little bit over and we still have a closing remark from Dr. An. So we're going to pursue to our closing remark by Dr. An, and also thank you for organization, I think Dr. An, Yang-Suk, and everyone who participate to organize this. This is really wonderful, and we should do this more often. And I will make a suggestion is, I think, we can share email address, and if people has additional question, I think we can all email each other to asking, and hopefully this will lead to many more collaboration in going forwards, either locally or internationally. So with this, thank you, and Dr. An, the podium is all yours.
Thank you very much for the Romney's the great the contribution to this meeting. So I think it's our first time to have Stanford and Korea, especially my team, to join together and then making some great scientific meeting, even though not in the contact meeting, but I think it's recently the Zoom meeting also is very educational, very important. So I hope we will have another opportunity to have some kind of scientific meeting, especially I first time to meet with Xian Ri and then James Zhang. So, and then also the Kwang Hyun I think it's really the great, and then to see the faces even though on screen. So this meeting is supported by the Korean Society of Academy of Science and Technology. So I think this Korean Science of Technology is really the outstanding Korean scientific committee. So I hope this meeting will be promoted in the future. Some more, many persons come together and then the great discussion. So thank you very much, all the persons to contribution to this wonderful meeting. Thank you.
Thank you.
Thank you.
We look forward more US-Korean collaborations.
Yes.
Yeah.